subject will be inheritance. So this is your actual genetics. Yeah. The whole unit, you know, inheritance and evolution, you think, oh, we're going to do a lot of actual genetics, but of course genetics is about which genes are expressed, how cells divide and all that sort of thing. So this bit is actually looking at genes being passed from one generation of cells to the next, or one generation of organisms to the next. And you've got some background notes which I'm not going to refer you to particularly for these, so they are for you to use. You've got some background notes generally on drawing of diagrams to illustrate what's going on, family pedigree diagrams. So this is rather like having a bit of a textbook. There's also another chunk of that book, the notes that we've been using, Mendelian genetics, in there, the monohybrid cross, sex determination, sex-linked characteristics, codominance. There's loads in here. A bit on sickle cell, not a lot. Lethal alleles, I must remember to put those in. Multiple alleles, and there isn't actually anything on dihybrid crosses in there, I don't think. But I'm aiming to get through quite a lot today. My plan is that we start off by looking at some of the terminology that we're going to need to use for this. It's very, very concise and precise. These words have very, very distinct meanings, and you need to be able to use them. Don't just try and describe something using everyday words. There are very, very clear bits of terminology. We've got words that we're already familiar with, like chromosome and gene, but then, you know, I've got a whole list of others to introduce. We'll then look at what Gregor Mendel did. Austrian monk in the 1800s, did a lot of breeding of pea plants because monks have time to do that. But yeah, we'll have some other examples as well. And we'll look at what happens when you get multiple options for a gene and how they're inherited, and then come back to the sex chromosomes, inheritance of sex, the important gene on there, sex-linked characteristics, and what happens in female cells, which is really interesting, I think. But then, yeah. Okay. If we start with the some of the terminology, if we have a pair of chromosomes, all right, so this is what we just described as a homologous pair of chromosomes. Doesn't matter which one they are. I'm not identifying the centromere on here. It's not an important part of what we're doing at the moment, although when you come to identifying the position of a gene on a chromosome, then it is important. So I probably should have included it. But along the length of a chromosome, we have sections that code for proteins called genes. All right, there's one. There's another one. There's another one. And usually you've got an average of about 1,000 genes on a chromosome, on a human chromosome. And of course a gene will always be, a gene for a particular characteristic or protein, really, will always be at the same position on the chromosome, and it will always be on the same chromosome. If it's not, then things go wrong. So the gene can be located. It's you know, it's a bit like having a map reference, you know, that you can actually find the position of a gene by going to a specific reference on the chromosome. And so we have the P's and Q's to represent the long and short arms. Well, we have the chromosome number, then P or Q, and then you have a number that tells you how far from the centromere the gene is. And we've already looked at that, haven't we? So that number, that location, is known as a locus. So that's at a specific locus. Okay, or location is what that means. So you can identify the position of a gene. Now, any of you that are doing a genetic condition for your presentation will need to show a diagram of a chromosome identifying the locus of the gene that you're looking at. Right? So, you know, that's a gene for a particular protein. Now, genes don't always come in the same varieties, and I'm going to choose probably the worst example I could, but it's one that people understand, which is the gene that controls the blue-brown eye colour. Now, eye colour is controlled by probably half a dozen different genes. They turn on and off different aspects of each other's function, and it's what will give you things like the greens and the greys and the mottled colours and all that sort of thing, is the interaction between these half dozen different genes. So just to say, oh, there is this one gene for eye colour is very, very misleading, but there is one that controls brown or blue. Now that means there is one gene for eye colour there. Let's just say it's the gene for eye colour. It isn't, all right, but it's one gene for eye colour. And it comes in two varieties, the brown or the blue. Now those two different varieties are not called genes, they're called alleles. All right, so they would be the two, maybe the same, maybe different, but these are alleles of a gene. So, how much terminology have we got there then? We've got chromosome, we've got gene, we've got allele, we've got locus. So we would talk about the gene for eye colour has two alleles, the blue allele and the brown allele. You can't talk about the brown gene and the blue gene, all right, because that doesn't make sense. All right, it's the gene for eye colour, and then, you know, there is a brown allele and a blue allele. Okay, now, there's a sort of list of terminology where we get, the words tend to come in pairs or groups. And if we look at those alleles, we can describe them in a lot of different ways. The words that are usually used are dominant and recessive, but there's also codominant, and you also get incomplete dominance, and there are others as well. Sometimes you'll get, rather than just two alleles for a gene, you might get 4 or 5, and you have almost like a hierarchy of dominance. So it's not just that one's dominant, one's recessive. You've got 5, and they might just go in a sequence, so that, you know, A is dominant over B, which is dominant over all the others, which is, you know, and you can actually, you know, and I will give you some examples of these. It's what we would call multiple alleles. But the idea of dominant, recessive, and codominant really is where I want to start with this. A dominant allele. Now, how do we think we might define a dominant allele? Well, yeah, we would represent it with a capital letter, but yeah, it's how we define it. Well, it does express itself, but doesn't a recessive allele express itself then? Yes, you have to have 2 of them. So a dominant allele, if we've got on this chromosome set up here, if we've got a dominant allele on both our chromosomes, yes, we would expect to see it. If we've got a recessive on both the chromosomes, we'd expect to see that. But the situation, if we've got a dominant on one and a recessive on the other, is where we get our definition from, because a dominant allele will be expressed even if there's only one copy of it. All right, even if it's on only one chromosome. So whether there's 1 or 2, you will see the dominant allele. So this is expressed whether on one or both chromosomes. Okay, so if there's 1 copy, you'll still see it. If there's 2 copies, you'll obviously see it. So that gives us a way of defining the recessive one, because that will only be expressed if, yes, you've got 2 copies, it's on both chromosomes. Good, good. Right, so this one only expressed when it's on both. Now, jumping ahead, this will give us a way of identifying what genes individuals actually have. Can you tell what genes somebody has got if they show the dominant condition? If somebody's got brown eyes, do you know what what alleles they've got? No, you don't, do you? Because they could have, because brown is dominant, they could have two brown ones, or they could have a brown and a blue. So you don't know what genes somebody has if they've got brown eyes. Do you know what genes they've got? Sorry, alleles. I do this regularly. Do you know what alleles they've got if they've got blue eyes? You do. So where you've got that situation, two recessives, you will always know, because that's the only way it can show. Now, this will give us some other words in a minute, but I just need to put in codominant. And this is where you've got both of them being expressed. So a good example would be, no, not in eye colour. There's no codominance in eye colour. If you get two different coloured eyes, you will always get a blue one. The blue eye is one where the gene hasn't switched on. Blood, yes, blood's the example. Which blood group shows codominance? AB, that's the one. You've got to think back to the physiology unit for this one. So this is where both of two different alleles are expressed. All right, so we'll come back to blood groups later this morning, but you have three alleles for blood group if we're just doing the ABO system, which is A, B, and O. A and B are codominant, so if you've got both of them, you show both of them. O is recessive. Okay, so back to that as an example later, but how are we going to describe individuals with these different combinations? First of all, you can have homozygous individuals. A homozygous individual will have two identical alleles. So two brown, two blue, two group A, two group B, two group O, whatever. So individual with two identical alleles. And so the other option is heterozygous, yes. Where we'll have two different alleles. Okay, now I'm not sure whether you'd describe somebody who's AB as being heterozygous. I suppose you would, really. So two different, yeah. Maybe you'd have to sort of qualify that a bit further, maybe. Homozygous, of course, you can have homozygous dominant, homozygous recessive. That's what I should have put in there. Heterozygous is just heterozygous. A couple of other words that come as a pair, because we've got three at the top there. We've got these two. The other two that come as a pair are genotype and phenotype. What we've been looking at so far is how we would describe the genotype, in other words, the gene combinations that an individual has got, or the allele combinations. So a genotype might be homozygous recessive. The other word that we need is phenotype, and that describes the physical appearance. So the phenotype would be blue eyes, brown eyes, group O blood, group A blood. Yeah? So genotype can be translated into phenotype. Genotype then is the genetic composition of an individual in terms of a particular characteristic, possibly. And then phenotype is the physical appearance or expression, if you like. Now, phenotype can be very confusing. you know, because you could have the gene, the alleles for brown eyes, but have blue eyes as your phenotype. Because if the genes don't switch on, you won't get the colour, and blue is no colour in eyes. And this is why if you have odd coloured eyes, one will be blue, because that's one where the colour gene hasn't switched on. All right. So the phenotype is really an interaction between the genome and the environment, and this is sort of where epigenetics comes in, because if you looked at those mice yesterday Monday, wasn't it, and you'd got the was it the big fat golden coloured one and the little thin brown one, then, you know, completely different phenotypes, and yet their genotypes were identical. All right, so but they looked different. The genes had been expressed differently. So phenotype can be a bit of a tricky one. We won't make it awkward as far as the exam goes. You know, if somebody's got a particular genotype, we will assume that translates directly into the genotype we'd expect. So we'll keep that straightforward. Now, the way that we write all this down, because we can't just write it out in words, is to use letters. And a lot of you will be familiar with the idea of using letters to represent genes and alleles. So it's the notation for this. We generally choose a letter to represent the gene, which is the initial letter of the gene, or possibly the initial letter of the dominant form of that gene, the dominant allele. But not always. It's not always obvious. For blood groups, for example, for the ABO blood groups, the letter that's chosen is I. All right. Now, I think that's because it's an immuno thing. It's not an immunoglobulin, but it's you know, the immuno antigens on the surface of the cell. I'm not sure what the rest of the word needs to be, but the I think, is immune something. But it's the recognition system. Yeah. So you have I with a superscript A, I with a superscript B, I with a superscript O to represent the three different alleles for that. But the I represents that aspect of the blood grouping system. The others might be quite obvious. I mean, the eye colour one, you don't choose E for eye or C for colour. You know, we do tend to choose B for brown or blue. It doesn't really matter for that. You have to be careful what letter you choose. There's no actual rule for this. You can choose what you like. But the way you've got to represent this is that the dominant allele will be an uppercase letter. The recessive allele will be the lowercase form of the same letter. All right. So you've got dominant is the uppercase or capital, or we end up saying big, and recessive is the lowercase. So, for example, if we take that eye colour example, which is appalling, and I will go through the P's thing with you in a bit. If we say the brown-blue eye colour, then big B represents brown, little b represents blue. We don't say capital B, uppercase B, lowercase b. We say big and little. All right, and that is just how it's said. Now, that means that you've got genotypes that can be expressed using those letters. So genotypes, I'm just going to draw a small table up here, and our phenotypes, just to use these words, and then I can go into the Mendel bit. For the genotype, you can have the homozygous dominant, which would be big B, big B. Phenotype, obviously, brown. Yeah. We can also, of course, have homozygous recessive, which is our little b, which gives us Blue. All right, those are fairly straightforward, but then the ones that confuse matters are always the heterozygous ones. All right, big B, little b. Now, theoretically, it could be little b, big B, but there's a convention that you always put the big letter first. It's a bit like algebra, to be honest. And when we start looking at dihybrid crosses, where we look at two characteristics at once, we then put them in alphabetical order as well, like you would in algebra. You know, if you're doing your multiplications in order that you can see that, you know, x, y, z is the same as z, x, y, and the same as y, x, z, you put them in alphabetical order, don't you? And it's the same with genetics. There's actually several algebraic similarities in genetics. So the phenotype for this one is, okay, brown. And yes, I had somebody yesterday saying to me, yeah, but my two grandparents have both got brown eyes, and several of their children, you know, parents' generation have got blue eyes, and then, you know, well, yes, they will have. You know, there's no sort of rules as to who gets what from their parents. You know, it is independent assortment, remember. But if you've got brown-eyed parents who have blue-eyed children, it's easy. We represent our two brown-eyed parents like that. All right? We know that if they have blue-eyed children, they must both be that. The way this works is like multiplying in algebra, where you multiply brackets together, left with left, left with right, right with left, right with right. Do you do the smiley face thing that you're all looking very blank? If you had, let's say, x plus y multiplied by x plus y, how would you do that? Yes, you'd do x times x, x times y, y times, wouldn't you do it like that? Yeah? You do the same thing. Oh yeah, the smiley face, I should have done it underneath, shouldn't I? Doesn't work like that. That's just an awful lot of eyebrows. You do the same thing in genetics. You do left with left, left with right, right with left, right with right, because these are our two grandparents, let's say, we'll just call them parents for now. They produce gametes that contain one of these. Half their gametes contain that, half their gametes contain that. Half their gametes contain that, the other half contain that. So we can now put, let's say, this egg with this sperm, and we get that. Yeah? We could, however, put that egg with that sperm and get that. We could take this egg with that sperm and get that. But of course we can put that egg with that sperm and get our blue-eyed. So two brown-eyed parents that have a blue-eyed child, that's how. It means that the parents are heterozygous, yeah. Or the gene hasn't switched on. Now, people misunderstand this. I'm sort of jumping ahead, really, into the next bit, the Mendel's peas bit, but people misunderstand this to mean that then if those parents have 4 children, they'll have 3 with brown eyes and 1 with blue. No, that's not what that means at all. It means every time those parents have a child, there's a 3 out of 4 chance of brown and a 1 out of 4 chance of blue. And it's the same every time they have a child. It doesn't mean that out of their children, that's what they'll get. All right, so you have to get this. The idea of statistics and probability and all this. We don't have probability in the maths unit, and I'm not sure how much probability they have in GCSE maths these days. Not as much as there used to be, I don't think. But there are all sorts of ways of expressing the likelihood of getting a blue-eyed child there. But yeah, we'll sort of, well, let's put that in here. I'm doing things in slightly the wrong order, but it's not that critical. So the chance of the blue-eyed child is what? Right, 25% or 1 in 4, 1 to 3, 0.25, a quarter. I think that's it. There's 5 different ways of expressing the same thing. You have to be careful. All right. I think that's all of them. If anyone thinks of another one, let me know. And, it just sort of depends what the question is as to what's most appropriate. Sometimes you might have to use the percentage or fraction to work out how many individuals in a population you might expect. And that's something we come back to next week with population genetics. Okay, well that sort of jumped ahead and given you this example. But I need to talk a little bit about Mendel. Gregor Mendel really did all the initial work that gave us this understanding, but he didn't know anything about genes or chromosomes or DNA or anything like that. So you've got the, there is this big section I put in these, these notes, these biology unit notes from, I can't remember what school they come from. It's down at the bottom, HGS, the S is school, and I can't remember what it stands for. But you've got this whole bit on Mendelian genetics. So the mid-1800s, Mendel was, breeding peas. But he had absolutely no idea, what was causing the different outcomes. Now, he was either just incredibly lucky, or maybe smiled on by God, being a monk and all that. But the results he got were phenomenally accurate to what we now know, how the genes would work out. Either that or he fiddled it. But, not not having an understanding of how the genes worked, I'm not sure that he could have fiddled it. But he looked at about 7 different characteristics in pea plants. Now, by almighty luck, I think, chance, whatever, these 7 different characteristics that he looked at were on 7 different chromosomes. You have a problem looking at the inheritance of characteristics that are on the same chromosome, because what happens in meiosis? First thing in meiosis, in prophase 1, you get, oh no, come on, crossing over. Remember, it mixes up the allele combinations, so they wouldn't end up being separated out from each other necessarily. They might travel together to one chromosome, or they might be separated to different ones. So if you have genes that are on the same chromosome, the inheritance will often show what we call linkage. You'll have genes that are linked on the same chromosome. But the ones that Mendel chose just happened to be all on separate chromosomes. I don't know how many chromosomes pea plants have. That's something to look up if you're really short something to do. But they were things like the colour of the flowers, the height of the plants, the shape of the peas, the seeds that were being produced, the colour of the seeds that were being produced. I can't remember what the other 3 things were. I think it was the position of the flowers, whether they were at the points where the leaves join, the nodes of the axis, or out. I can't remember the rest of it. But he looked at the inheritance of all these, and he found that, I can't remember the word that he used, and it's not in there, but he looked at what he called traits. Trait is another word for characteristic. So, just replace one with the other if you like. But these, it says here, he investigated simple qualitative characteristics or traits. So his list, flower colour and seed shape, that's all they put. But he was lucky really that these characteristics were controlled by single genes, and those single genes were on separate chromosomes. You know, that is just, it's what made all the difference to his studies, because other people had tried to do breeding experiments looking at characteristics that we now know are perhaps controlled by several genes, so you can't follow a clear inheritance. Where you've got characteristics controlled by a single gene, it makes life so much easier. All right, so things like ABO blood groups, for example, are easy to look at. Just trying to remember the ones that we list when we're doing the GCSE biology. There were, I think, there were 4 characteristics I used for people to look at. One of them is earlobes. Earlobes are either loose, like mine are, or attached, where they go straight in. Yeah, and that's a single gene characteristic. There's, if you've got a bent little finger, and I can't remember what they call it, but it's you know, where the little finger bends out, is a single gene characteristic. The ability to roll your tongue is a single gene characteristic. Yeah, all right, so Wendy can't do it. So, you've got the thumb, yeah, hitchhiker's thumb they call that. Yeah, now I've got the straight one. But yeah, the tongue rolling, that's a dominant characteristic, so we know you are homozygous recessive for that. It's, you know, you could pay, what is it, about £150 and send off a sample of your cells to get analysed by, you know, these big companies, or you can just work it out. But you don't get the really interesting stuff. Oh, pea plant's got 14 chromosomes, yay! Yeah. It's, yeah, there's no real sort of link between numbers of chromosomes and anything. The other thing, and it's funny, I was just talking to somebody at my Tai Chi class about this last night, because we were doing an exercise. Apart from the actual Tai Chi sequence that we learn, we also do other stuff. There's swords and there's fans and there's sticks, and we were doing sticks last night. Sticks is good fun. But we do some exercises where one person holds the stick and you sort of have to move it about, and you're sort of, you know, trying to stop the person attacking you with a stick, basically. But it was how we were holding our hands on the stick and which hands we were tending to use. The lady I was working with is an ex-GP, and we were talking about this, and I said, one thing that we have on the GCSE that I use, which I'm not convinced is, all right, just do that with your hands. Right, which thumb's on top? Right, mine's the right. Yeah, yeah. Now they say that's genetic. Now I think that also associates, yes, with crossing your arms as well. Right, you've got it that way. You try and do it the other way. How wrong does that feel? It's completely wrong. No, it's actually you've got to completely. You have to completely take them apart and put them. And it just feels complete. Yes, it doesn't feel right. But it's the same with crossing your arms. You know, if you do it one way, you try and do it the other way. Yeah? I mean, I can't do it anyway. Yeah. But these are likely to, you know, you can actually ask within your family, you know, who's doing it which way, and it's something that you can find out for yourselves. There's another thing that you can try. Bear with me. Okay, looking at what Mendel did, we have this whole area of genetics now. It's just called Mendelian genetics, and it's this ability to predict, really, based on certain rules. And Mendel's peas are, you know, the classic that we go back to. And I'm going to just take an example. We'll look at pea colour and pea shape. So, yeah, Mendel's crosses, really, is what we're looking at. First of all, if we look at the idea of the monohybrid cross. Monohybrid means a single characteristic. And we're going to take, for example, pea colour. So, dominant colour in peas is yellow. All right. And so we represent the green we're more familiar with a lowercase y because that's recessive. So why are all our peas green? Yes, exactly. We've chosen to breed them that way. We prefer green peas. What's the genotype of a green peed plant? There's no G's involved. The genotype of a green peed plant will be little y, yes. So if we only ever breed together plants that are little y, all we'll get is little y. And that's what we're doing. Oh, that's odd. Oh, for pea pods. Yeah. Okay, what's G representing though? That's a curious one. Okay.
So big G for green pods, little G for yellow pods.
Okay, I haven't come across. Right, the pod colour is not something I've come across. That's a curious one. Yeah. Okay. Well, that you could say about anything. In terms of colour, what you see is the light that's reflected. So if you want to say, you know, what colour's Wendy's top? It appears to be pink because it's reflecting pink light, if there is such a thing. Although this leads me on to a strange debate we had at home for a long time. Is there any such thing as brown light? You see, and we decided, no, I looked it up and there's no such thing as brown light, which means how the heck do you see brown things then? It's combinations of other colours, yeah. It was when we were sort of, I don't know whether it came from Red Dwarf or something like that, where they had to change the colour of the light bulb. If they had an emergency, they had to put the red light on and change the light bulb. And I said, well, if it's a real emergency, you have to put the brown light on, don't you? And that set us off. So can you actually get brown light bulbs? And yeah, but no, what you see is the light that's reflected. So that's not actually the colour of the thing, is it? But it's how we define the colour of the thing. Sort of, yeah. But we define the colour of something by the light it reflects. So the other thing with leaves, though, is that most of what's reflected is green from chlorophyll. The chlorophyll obviously absorbs red and blue light, but you've also got a lot of other pigments in there as well. There's carotenes, which are yellowy orange, xanthophylls, which are sort of browny reds and things like that. So they do get a bit complex. Anyway, back to our peas. Now, what Mendel did, Mendel's first cross was to take, and he did this with all these different characteristics, he took pea plants that he described as being pure breeding. Now, to us, that would be homozygous, but he didn't know that at the time. He just used plants that for generation after generation after generation of interbreeding had produced the same characteristics in the offspring. So he crossed a pure breeding yellow with a pure breeding green, which we now know to mean a homozygous yellow with a homozygous green. And if that's our yellow and that's our green, then if we do the crisscross way of doing this, I'm not going to keep putting all the colours in. Hopefully you can see that, you know, this plant can only ever give a big Y, that one can only ever give a little y. So we're going to get all the offspring will have the same genotype, which is heterozygous, and colour-wise they're all yellow. Yeah. So all heterozygous yellow. Now, I've not really put very much information in there. I should have put the gametes in probably, but sort of saving space here. Now, this first generation after these two homozygous parents is known as the F1 generation. This is the F1 generation. F standing for filial, which means sons. F1 generation. Yep. So an F1 generation comes from two homozygous parents. Sometimes, if you're a keen gardener, you will buy F1 plants. They call them F1 hybrids. Reason being, they will often have been bred to have particularly strong characteristics from two parents who weren't individually necessarily strong, but by bringing them together you can make strong offspring. So the F1 hybrids are very often stronger than either of the parents. The trouble is, if you then try and plant your own seed from crossing two F1s, you'll find out what happens in a minute, because you revert to some of the earlier genotypes. And when people talk about a certain characteristic skipping a generation, this is what's happening. So you know, you could say, well, there's no green in there, so if that turns up in the next generation, it's skipped a generation. Yeah, you can say like that if you like, but you know, describing the genetics is a bit more meaningful. The other way of representing this that some people prefer, and you'll find it in some of those notes sheets, is to use a square. That if you take one parent up here, the other parent over here, and you put their gametes like that, yeah, a lot of people seem to find this easier. And you then just put the combinations together, reading across and down. So you've got the two big Y's there, a big Y and a little y, a big Y and a little y, and two little y's. Now, this is a simple form of what we call a Punnett square, which we will come on to next week for doing dihybrid crosses, where you've got to do two characteristics at once. It's the only way you can do it. So that's just an alternative way of doing it. And we have the same thing in terms of, you know, talking about the probability of getting any of those genotypes. Okay, now the next bit then was Mendel's second cross. Oh, hang on a minute. No, it's all right. Mendel's second cross was two F1 individuals. In other words, two heterozygous ones. And this is taking me back now to our two brown-eyed parents who had the blue-eyed child. So hopefully you can see what we're going to get this time is if we take our two heterozygous yellow individuals, we get the same process, and it is a bit bad leaving out the gamete layer in this. Oh, that's what I just drew. Oh, sorry. I was just thinking, why do I remember drawing this? Sorry. Yeah, that's the second cross then. I should have done big Y, and big Y, little y. Sorry, big Y on one side, one edge, and little Y on the other, shouldn't I? Right. So yes, that actually comes with this one. My brain was ahead of... No, my hand was ahead of my brain, wasn't it? Right, we put those together. All right, so we then have one homozygous dominant, two heterozygous, one homozygous recessive. But what's key here is the phenotypes. So how are we going to describe this? The overall effect is best described in a ratio 3:1. Yeah. So these are yellow. That one's green. And so it's 3:1. And that there is known as the Mendelian ratio. Three to 1, okay, which is what you get if you cross two heterozygous individuals. Okay, we will sidetrack a little bit just before we go to break onto multiple alleles, because this is all very well. You could do the same here for the shape of the peas. Are they round? Are they wrinkled? Round is dominant over wrinkled. You could do it over the height of the plants. Tall is dominant over short, or dwarf, they tend to say in plants. So you could do this with, you know, any of the individual ones, but they're all just controlled by single genes with two alleles. So you don't achieve anything different each time. And this applies to quite a lot of the genetic conditions that people are going to be looking at. That it is quite a simple single gene with two alleles. Sometimes there are different forms, different mutations perhaps in genes. Cystic fibrosis is an example of that, where, yeah, it's one gene that's involved, but there are several possible different mutations in there. And yeah, so we know that at the protein level and the actual cell function level, this is going to be a lot more complex than just having these alleles. But what if we've got more than one? Sorry, more than two alleles. Multiple alleles as a sort of additional bit on this. As an example, we'll look at blood groups, but there are plenty of others. So if the gene is I, the alleles we have are I with a superscript A, which will give us the, if you remember, it was a particular sugar sequence on the outside of the red blood cells. I could go back to that, but there was like a chain of sugars, and the terminal sugar was what decided the blood group. If there was a chain without, I think there was a chain of 4. If you didn't have a fifth one, it was O. If you had a certain fifth one, it was A, but a different fifth one, it would be group B. There were also these rather unusual individuals who don't even have the 4 that you get in O. And that's the Bombay blood group, which, Bombay blood group, yeah, first discovered in what's now Mumbai. Of course, they haven't changed the name of it, but yeah, it would be good if there was enough on the Bombay blood group to be a presentation topic, because I think that's really quite interesting, because those people can't even have group O as a transfusion, because they haven't got anything on the outside of their cells. They haven't even got the O bit. But they don't come into this, all right? Genetically, they are incredibly rare. So your alleles are the I with the superscript A or B, and those are uppercase because they are codominant. The other option is an I with a lowercase o, because that's recessive. Okay, so these are codominant. Now, we've got, from these three, then we've got various possible genotypes, and from that, the phenotypes, haven't we? So, let's do ourselves a little list. Okay, well, let's start with the most obvious. If we've got IA, the phenotype is A. The other one involving A that's going to give the same result is IA, IO. Yeah, so that will give us A. We've got the same thing with group B, that if you're IB, your group is B. And also IB, I, O will give you group B. Now that means we have IA, IB. IA, IB will give us group AB, and that just leaves IO, IO. Thank you. I can relax now. It's, the thing with this job is, you know, at certain points in the year, you know you've got to say certain things. That anticipation has been building in me all morning. And I will not write that list in any other order ever. All right. And yeah, some years are better than others for the response I get, but yeah, I owe. Is the only way of getting group O, is what you should say, of course. But yes, so, you know, from that, of course, we can see that there's, you know, no particular link between dominance and, you know, how common something is, because O in this country is the most common blood group, but of course it's the homozygous recessive. Whereas A and B, you know, group AB is the rarest one, and they're codominant, so there's no link. But again, I think when we did the bit on blood groups, I showed you some maps of the world and how the alleles were distributed. You can't remember that, can you? No? Oh, right. I was going to see if I can find that just to finish this little bit off. Trying to think where it would be.
I know one. I know this to be A. I know the phenotype. When you get together with me, you will know.
Oh. We had these maps, if you remember. These should now make a little more sense. If we're looking at the distribution of group O, well, I'm not going to run it as a whole presentation, but if just to remind you, it was about these little chains on the outside of the cell. So O had the 4 sugars, A had those 4 sugars and another one, B had those 4 sugars and a different one, AB had some of each, and of course then you've got this Bombay blood group that doesn't even have what the O's got, which is a, you know, it's a curious thing. Anyway, going back to those maps briefly. Yeah, group O, most common in the pale areas and into the orangey reds, and then, oh no, sorry, other way round, isn't it? Least common in the pale areas and the orangey reds, most common in the blue. So group A, sorry, it's not group A, it's the A allele. So that should now make sense that that is in group A and AB. So it's, you know, it's a little harder to track that. Most common in some rather odd places, sort of up Alaska and top end of Canada. Stripe of it going through Australia there. Australia is quite curiously sort of patterned, isn't it, for that? And then odd little patches of light blue in the top end of Scandinavia. I never know which bit's which up there. And then, yeah, the B allele, incredibly rare in the Americas and Australia. And then almost a pattern that, you know, gives the greatest density, you know, in quite a distinctive stripe down the middle there. So it's, yeah, how up to date those are. You know, the thing is with movements of people, obviously this changes, doesn't it? And, yeah, we're just looking at patterns, really. But there are links between blood groups and resistance to certain diseases evolutionarily, so they will have been selected in certain areas for certain reasons. Those are not necessarily reasons now, but you know, that's the way blood groups go anyway. Right, that's a good place for a break, I think, isn't it? Moving on then, the next main bit I'm going to go on to is about sex chromosomes and things, but it just crossed my mind to put something else in while we're in the monohybrid section. So we've had this bit on multiple alleles. What you have to bear in mind is that although we talk about these genes being genes for a characteristic, a gene is only ever a code for making a protein. All right. Now, in this case, you know, if you think about what this means for blood groups, and I'm talking about chains of sugar molecules on the outside of a red blood cell, how does the actual gene code for that blood group then? What do you think it might code for? What's the most common sort of protein in a cell that's going to do a job? Begins with an E. Enzymes, thank you. Majority of genes actually code for enzymes that do jobs which then appear to give a result. So, for example, in the blue-brown eye colour, we've got an enzyme that's responsible for the production of melanin, which is the brown pigment in the eyes. Yeah. With this one, the A and B will be coding for proteins, in other words, enzymes that are responsible for adding different sugars onto the end of the antigens. All right, so it's only ever about the production of a protein. So it can be a bit misleading, but again, you know, if you're looking at a genetic condition for, you know, your presentation, you've got to be looking at what does, you know, what's the normal protein that's produced by the gene, what happens when the gene is in a, you know, mutated or damaged form or disease form, what happens to the protein production or other aspect of gene expression. All right, and it's not that, you know, these genes aren't responsible for directly for those antigens on the surface of the cells. They are responsible for enzymes that will add sugars to the antigens. While we're on this topic, though, just looking at the sort of more unusual sort of examples, perhaps, just going to stick something on here to do with lethal alleles. Let's say, for example, that you crossed two heterozygous individuals and you only got offspring that showed the dominant characteristic. So let's say, I'm not choosing a particular example here, e.g. if you cross, I don't know, choose a letter we haven't used yet, H. All right, but you don't get any individuals, so no offspring show the homozygous recessive condition. So, you know, what's happened there? This leads us into not a direct question on the exam, but it's related to a question on the exam, because it's not necessarily as obvious as this. But if you've got these two individuals, then you would expect, if you cross those, you'd expect to get a big H, a couple of, whoops, slipped there. I've just crossed the same thing. Start that again. Right, a big H, little h, another big H, little h, but you don't get that one. Why not? You know, if let's say these were plants that were churning out hundreds of offspring, but none of them seem to show that characteristic. It's because of this idea of lethal alleles, that particular genotype is lethal. Okay, the little h in this case is a lethal genotype. Now, you're actually given an example in your handouts. There's one to do with Manx cats. Now, what's characteristic of Manx cats? Manx, from the Isle of Man. Manx cats. Got no tail. But it's not that they've got no tail, particularly. They have an incredibly tiny bit of tail. And if you look, if you find those notes that came from that A-level source, there's a page 80 that says lethal alleles of it. This is it's a codominance example, actually, if you have a look there. Manx cats, they appear to have no tail, but they have just a little bit of tail, and this is a genetic thing. Now, the problem is, of course, the tail is part of which structure in the body? The spine. Yeah. So what's happening here is there's a shortening of the spine. Now, there is a combination of alleles which causes so much damage to the developing spine that it's lethal. So if you look at the example you've got there, if I do it the way that I've been doing it here, with Manx cats, normally you've got this codominance situation. Let's just get this right. There are two alleles. You've got, let's just check them out. S with a superscript N is the normal allele, and SA is absence of the last few vertebrae. All right, so SN is normal, SA is absent vertebrae. All right, the last few that make up the tail. But this is a codominance lethal alleles thing. And so your options are that you can have a normal combination where you've got SN, SN. All right, so you get your normal tail. So, you could cross two Manx cats and actually get one with a normal tail. If you cross, if you get the heterozygous condition, you've got either that or, of course, the other option, I need to put that in occasionally. That's your Manx with the short tail. Doesn't appear to have a tail. If you were to X-ray the spine, you just have a few there. But if you look what you've got, there's the other option is the SA. And so that's the lethal combination, and it tells you in here the spine doesn't develop, so this is fatal early in development. The embryo doesn't develop and is absorbed by the mother, so there's no evidence of its existence even. So you never even know about it. It's not that you get kittens with no spine. That the development is, that because that's such a critical part, that bit doesn't develop. The cells are recycled by the mother. Embryo doesn't develop. Now, there are other examples of genotypes where you've got life-limiting combinations. And there are impacts perhaps on the number of individuals in a population who can actually pass something on. Now, I don't want to go too deeply into, Huntington's is an option, but that's Sonia's topic, because that comes on later in life and it does kill people. Then you'd think it would restrict the amount that it could be passed on, but obviously if it comes on later in life, then people may have already passed it on, and there's all the issues associated with that. We'll leave that one alone. But there are others, and there are genetic combinations that can, for example, cause children to die in infancy. So rather, they are still lethal, but I'm not sure whether they have a different description. But they're not lethal, before birth. So their genotype appears in birth statistics, if you like, but they don't survive to be able to pass it on. So when you actually look at the next generation, you may get fewer of those individuals, and it may appear in the adult population that there's fewer of those particular genotypes as well, because they haven't survived. So, you know, there's other examples, and there is an example along those lines in the exam. Right? So other, they're not really examples per se, but other examples, I'll put Huntington's without saying any more about it. And then we've also got diseases. Oh no, I shouldn't put diseases, should I? I should put genotypes. So in infancy basically means sort of before puberty, before reproduction, so that those alleles then don't get passed on. And it's sort of evolution, I suppose. It's natural selection that if those genotypes don't contribute to survival, then we lose them. You know, that's the harsh sort of biology of it, isn't it? So yeah, it's not always that a phenotype is going to appear. All right, so sometimes you will actually lose them, and yeah, I've seen example questions where you've got the ratios are not right, and that will be why. You know, examples of the statistics not matching up to what you'd predict. Right, so where have we been? Let me just check back. We've done the monohybrid, so the first cross giving us all the heterozygous ones in the F1 generation. I then, put the wrong square in there. The second cross between two F1 individuals, I have missed something out. That's right, I should be calling these the F2 generation. I had a feeling I'd gone through this too quick. And then we get from this the Mendelian ratio, that if you cross two individuals who are heterozygous, you will get a 3-to-1 prediction of phenotypes. And 3 showing the dominant characteristic, 1 showing the recessive. Of course, you know, confusion sets in when you start looking at multiple alleles. And yes, it leads to all sorts of questions, that one. And then we have this possibility of lethal alleles where you actually lose individuals either before birth, so they never actually appear in the statistics at all, or they're lost in infancy where they don't make it to reproduce. So you know, the number of individuals in a population with a particular genotype will probably be, you know, well, it'll be less in terms of the disease or more in terms of the normal condition. You know, it will appear that it's less prevalent than it is. Okay, now that then leads me on to sex. Yes. So now in mammals, the sex chromosomes are the X and Y, and we all have an X chromosome, but it's what else we have that makes the difference. In other animals it's different. In birds, for example, it's not. They're not called X and Y, they're called W and Z, I think. And it's the males that are homozygous. Can't remember whether they're WW or ZZ. I think they're WW, and the females are WZ. So, you know, it's not the same in all organisms. So, you know, it's just that these are a separate pair of chromosomes. They're in humans, obviously, it's our 23rd pair. The rest are called autosomes, and so sex is determined by the combination of sex chromosomes that we have, theoretically. they are chromosomes which can have an awful lot go wrong with them and yet still be survivable. Hence, we do get quite a lot of, well, you get this idea of intersex, don't you? Yeah? There was, so I mentioned it recently, there's a model, isn't there? And bear with me. It will take just a minute. She was in the news. Right. Now, you might look at her and say, well, yeah, she's sort of got characteristics that are almost between male and female. But the default development is female. But it's, you know, you could equally say, you know, that's just family traits and, you know, sort of perhaps of quite a square jaw, that sort of thing. But, yeah. So to all intents and purposes, she's female because she, you know, has an active X chromosome, but she also has a Y chromosome. It's just that the Y chromosome isn't doing anything and never has. But there is a whole sort of story around this that she has. She's Belgian, revealed she's intersex. She's gone public in an attempt to reduce the stigma around the condition. She was born with internal testes and without a uterus or ovaries. And she has a condition which is one of the most common issues with sex development, which is androgen insensitivity syndrome. Androgens are male hormones. Now, what you have to have in order for a human being to become male, you have to have that little Y chromosome. Well, you don't actually have to have the whole chromosome. You have to have the SRY gene. That then has to interact. Now, we know about genes and hormones interacting, don't we? The body has to be, the developing body has to be sensitive to male hormones in the mother's body. Now, if the fetus isn't sensitive, if they have this insensitivity syndrome, the mother's male hormones don't trigger the activity of this SRY gene, so the embryo doesn't develop the male root. And although it's got an X and a Y with an SRY gene, it doesn't work, doesn't function. So, you know, when we're considering what I tried to do on Monday, not very well, about control of gene expression, this is actually a really good example. Androgen insensitivity syndrome is where the SRY gene is not expressed because of an issue with it. But it's yeah, it's, you know, she's not the only one by any stretch of the imagination. So you know, it's not as simple as male female, but we sort of have to assume it is, don't we? So we've got XX in humans is our female, and then XY for the male. So it is fathers who determine the sex of their children. Not that Henry VIII would have had that, of course. You know, and of course, you know, you go back into history, of course, you know, where males were required. It would be seen to be the mother's fault, but yeah, before people knew about the chromosomes involved, of course, you know, you've only got to do one of those and you can see that you get your 50-50 or, you know, 0.5 chance of either. All the egg cells, of course, from a woman will contain X, at least one, possibly more. Problems if there are more than you need. If you look at the father, though, obviously you've got two different types of sperm, X sperm and Y sperm. Now they are slightly different. Which would be heavier? The X, yeah, because the X chromosome is bigger. You know, we're talking about fractionally, aren't we? But the X chromosome is quite a lot bigger than the Y chromosome. That is enough to actually separate them. Now, I don't know if they do this for humans, but I know they do it in cattle. If you know cattle breeding, you can artificially inseminate cattle. We won't go into the whole procedure. I once I had a group who insisted we find out more about artificial insemination in cattle. But yeah, it's yeah, well, it was something we wished we hadn't by the time we got to the end of it, I think. But that was it was the collecting process that was the yeah. Oh right, just sort of yeah. You have a sort of fake cow. Anyway, never mind that bit. But if you collect the semen from the bull, it can be put through a machine that will sort it into X and Y, X and Y sperm. Now, the reason that's useful is, of course, if you want beef cattle, you want to be producing males, and if you want a milking herd, you want females. So because otherwise you can get an awful lot of waste of calves, can't you? Because it's not necessarily the same breeds, varieties of cattle that are good for milking and meat. So you can get a lot of waste. They are basically disposed of or used for veal or whatever. And so it's, it can be used. So you can actually differentiate between the sperm, male and female sperm, essentially. All right, or the X and Y sperm. Theoretically, they could do it in humans. I don't know if they do, but because certain conditions are carried on the sex chromosomes, you might have a situation where, somebody wants to have a girl so they don't carry a sex-linked condition through the family. So theoretically it could be done, but what they tend to do, which I think is easier, is just to make a lot of IVF embryos and then analyse them at the 8-cell stage. And using gene probes, of course, you can easily flag up whether there's X or Y chromosomes there. Anyway, what we're looking at here, of course, is whole chromosomes. All right, these are not genes. So if we actually want to look at the genes that are carried on these chromosomes, then we, you have to bear in mind the difference between them. And I had... Sorry, just have to click back. I should have opened this in advance, shouldn't I? But back in that, the chromosomes one, the one that starts with my shoes. Remember the shoes? But I gave a few images of chromosomes at the end. I wanted the X and the Y. That's the one. Let's just show it from that one. I don't think I actually gave you these. But the difference, you have got those, yeah. Okay, so you've got those somewhere in your handouts. The difference between them is huge. Now, the problem is that, these are disorders that are mapped out on here. Obviously, these are what happens when certain genes malfunction. So these genes have normal functions. Yeah, so you have to remember that you think, these chromosomes are full of genetic diseases. Is that what they're for? Well, no. You've got to remember that the normal forms of these genes actually do jobs. Now, the problem as far as the Y chromosome is concerned is it doesn't have all the genes that the X chromosome does. So if you've got an X and a Y, then you've got only one copy of certain genes. All right, and of course, if that includes certain conditions, I mean, the classic ones that are carried on the X chromosome are things like haemophilia, red-green colour blindness, the ability to light a barbecue, and the ability to change channels so many times with the remote control you can't keep track of where you are. But there are quite a lot of conditions that we associate with males, not females. Duchenne muscular dystrophy is another one. But the story is a bit more complex than that. It's not just down to being male or female, because the thing is with this X chromosome, it's a big one. It's very powerful. It's got an awful lot of content. It's got a lot of very influential genes on it, let's say. Now, the problem with that is that, all right, in a male, you've only got one copy of this X chromosome, so anything that's on that X chromosome is likely to show, regardless of whether it's dominant or recessive. The problem for females is that actually 2 X chromosomes is too much. All right, there's too much activity from a pair of X chromosomes. So what you get in female cells, somewhere in development, and I've forgotten whereabouts it is, I think it's at the point of about 500 or 1,000 cells or something like that. In every cell in the female embryo, one of the X chromosomes switches off randomly. But every cell that then develops from that one has the same one switched off. So what you end up with are patches of cells, really, in the female body. That some will have one X switched on, some will have the other X switched on, and the other one switched off. So we don't have the same X chromosome functioning in all our cells. Except that, of course, you could do. If it's random, there is that option, isn't there? You could actually have the same one operating in all the cells. So I'm going to come back to that. I'm going to start with this idea of sex-linked disorders. And need to choose one. Just check who's doing what. Right. Yeah. I'm gonna try not to step on Taylor's feet because she's doing haemophilia. I'm going to choose red-green colour blindness for them. This is a problem I always have at this point, that I have to, you know, what I might be doing, I then have to check that I'm not going to actually teach somebody's presentation for them. Red-green colour blindness, which I think is that one word? Yeah, might be, might not be. Don't think it is. Yeah, males are. That's what this is about. Yeah. There are different sorts of colour blindness, but the red-green is much, much more frequent in males. Normally I would do haemophilia, but I'm not going to. Just a quick outline of haemophilia. Haemophilia is due to a fault in blood clotting. And it is a recessive allele, but so usually women will have enough of they'll usually be heterozygous and have enough of the dominant normal ones switched on not to be a problem. But males, if they've just got the one copy of it, yeah, tough, you know, they'll have it. So I won't say any more than that, apart from Queen Victoria. You will mention her, won't you? The way that we represent sex-linked characteristics is we have to tag them to the chromosome. And colour blindness of this type is where the cones that recognise red and green don't differentiate between the two wavelengths. So they see a sort of intermediate, I suppose, almost brown. So yeah, there's me saying, you know, oh, you can't really see brown. It's a sort of reddy green, which is pretty much brown, isn't it? But if we take, let's take C for colour. If that's normal, then and that's colourblind, then our alleles, well, the way that we would represent them, the possible genotypes that you could have for a female, of course, you can have, whoops, C is not a good one to use, I just realised. All right, you can have that. All right, you can have a woman who's got the two normal alleles on her X chromosomes, and it doesn't matter which one switches off, you see, she'll be fine. You can have the heterozygous, in which case, yeah, possibly half her cells have switched off the good one, but the chances are there's enough functioning normal X's left to actually be okay. The only way you get women with red-green colour blindness is the homozygous recessive like that, which is just much more rare. But of course with men you've only got the option of that or that, because they don't have that gene on the Y chromosome. All right, so you'll have some normal, some affected. So that's the one that, you know, can't cover it up like this one would. That's the equivalent to that, but this one, yeah, you know, you don't have that option. Another really good example would be, there's a really interesting example. No one's doing muscular dystrophy, are they? No. I have to keep the list at hand all the time now. Duchenne muscular dystrophy is a condition that's X-linked, and it causes wastage of the muscles, and is usually fatal by teenage years. You know, it's just a gradual wasting disease, so it's a sort of atrophy of the muscles, or dystrophy is what it's called, isn't it? And it's down to the production of a protein in the muscles called dystrophin, which I think it sticks the muscle fibers together or something like that. And there was a really interesting story, if anyone wants to change their mind and do this one, you can do, about twin girls who had muscular dystrophy in the family. Now, normally, girls wouldn't show muscular dystrophy because half their affected X's would turn off, half the unaffected X's would turn off, but they're left with enough functioning X's to be unaffected. But there were a pair of identical twins, one of whom had muscular dystrophy and the other one didn't. And that was because, you know, although we think identical twins are identical, they quite clearly aren't, because what you don't get is the same X chromosomes switching off. And in one of those twins, far more of the normal X's had switched off, leaving her with muscular dystrophy, and in the other one, far more of the muscular dystrophy ones had switched off, leaving her with normal muscles. So identical twins, one affected by a really serious life-limiting genetic condition and the other one not. So again, you know, when you think about the control of gene expression, just the fact that these X chromosomes can switch off is a factor, isn't it? So, you know, there's more and more fascinating stuff here, isn't there? And, you know, you start thinking, well, do I want to change my subject? And don't. I don't know whether I've given you, no, I don't think I have, but I will put it on, I will put this on Moodle. I have PowerPoint to go with this. Actually, no, it really wants to go on the bottom here. We have this process called X inactivation. It's also known as lionization. Presumably it was elaborated by someone called Lion, and this is this process where one X in every female cell switches off. It becomes visible in the cell as a little condensed structure called a Barr body. So one X in each female cell switches off. And condenses into Barr body. Now, this book that I promised you I'd read before we got to this unit, which I didn't, and apologise, and I'll be promising next year's group the same, I'm sure. Trouble is, trying to read it at bedtime, it just wasn't working. There was a bit about this process, and it's all about this idea of junk DNA, or DNA being a lot more flexible than we ever thought it was. And this process of X inactivation is controlled by a gene, that's read off both strands of the DNA. So I just remember little bits, because the trouble is, when you read stuff before you go to sleep, you know, some of it sticks and some of it doesn't. So I can't really tell you anything about the details because it's not stuck in my mind properly. But it's controlled by a gene that's called X. I can't remember if there's a hyphen in it. I think there is. Xist. It'll be X, yeah. Well, it's X inactivation something something. No, that's not right. Yes, it is. Yes, it is. But on the non-template strand of the DNA, you also get the same part of it red, but obviously it goes the other way. And they name the other part of this T6 because it's read back the other way. But it's this gene on both strands of the DNA. It's weird, right? But it's fascinating. But I clearly have got a lot of reading to do. But, you know, if you're going into anything that's related to biological sciences, this is the sort of thing that you will come across. But, you know, we're finding out an awful lot more about these control systems. But the best example of this X inactivation, the lionization, is in tortoiseshell cats. So let's show you this. This you haven't got, and I will put this onto Moodle. And you haven't got any notes on it either, I don't think. All right, there's the exist. It didn't have a hyphen in it. This is the chromosome that's going to be inactivated. And basically, well, you've got, I think HP is histone proteins would make sense, wouldn't it? Because you've got, if you think, you have this normal sort of folding of DNA anyway using the histones. And this exist gene is responsible for triggering the compaction of the second X chromosome, right? So it appears in a cell. You can't really see it over there. Hang on, if I can draw around. There's a nucleus actually in here, sort of like that. Okay, and using fluorescence, they've managed to flag up this little thing at the top, which is now a bar body. Right, that's the bar body being shown up there. But this inactivation process triggered by this this special gene that just folds it up on itself and tucks it away at the side, so it doesn't work anymore. You can't. I don't know whether you can see them on there. This is the trouble when you do a presentation and you look on your actual laptop or monitor screen in front of you, and you can see these things, and those are fairly evident on here, but they are just these little condensed blobs inside the nucleus. And it helps you identify female cells without actually having to look at the chromosomes. Right, if you can see a bar body, it's a female cell. Right, so what happens? Well, you start off with a female zygote, a fertilized egg cell that's got 2 X chromosomes. Now, this is leading to the tortoiseshell cat example, because tortoiseshell cats carry a gene on the X chromosome for coat color. It's a codominant one, and it comes in 2 alleles. You've got the black and what we'd call ginger, but technically is called orange. Hence the 2 colors on here. So we've got a zygote where you've got one X carrying a black allele, the other carrying the orange. Now, in the early embryo, we've just got 8 cells, 2, 4, 6, 8 cells there. You can see that in some of the cells, the X carrying the orange has switched off and become a bar body. In others, the one carrying the black has switched off and become a bar body. So each of those cells will now divide to produce cells with the same X inactivated and the same one activated. So you get patches of black fur, orange fur. All right, so they continue, once you've switched off one of them. These have switched off the black, so they all produce orange. These have switched off, we've got a mixture there, so we get the patches of cells. And if you follow them through, this all got a bit overcomplicated once I started adding more diagrams in, but depending on which one switched off, you will get very different sort of patterns. And so you get that sort of effect. All right. So, a tortoiseshell, therefore, will only ever be female. Yes, you. No, they're not. No. A ginger female is rare and quite valuable. Yeah, ginger females are quite rare, but because you could have 2 X chromosomes both with the orange, it's just that they are quite rare. All right, there are many more black cats around. That's the thing. So yeah, so tortoiseshell cats have these patches of orange and black, and they are usually a bit psycho as well. These tortoiseshell ones are usually a bit crazy. Yeah, yeah. All right. Yeah. Well, my son and his girlfriend have one who she is absolutely nuts. Then turn and hiss. Yeah, that's what this one does. They're very unpredictable, I think. So you get that. I think they're absolutely beautiful, but yeah, unpredictable. Now, depending on whereabouts you get the cells switch their X chromosomes off. I mean, that's quite a neat pattern. But how about those?
Yeah, well.
Yeah. All right. Now, the one here on the left is called Venus, and she has her own website and everything. And I don't know. The other one is obviously a different arrangement. But you can if you think about how the embryo works now, you can see how that's happened. But the difference is astounding, isn't it? These just like two cats glued together. That you've got, I mean, eye colour as well, obviously, is related to that gene. Must have something to do with it, mustn't it? Perhaps it's next to it or something. But given that it's an enzyme thing, so yeah, the black cat half has got the green eye, and the ginger cat half has got the blue eye, and then the rest of the body is just tortoiseshell. I'm not sure. Yeah. I don't know how far that colour definition goes, but yeah, it's because of course bodies develop symmetrically, don't they? So presumably, you've got the switch off is before that happens, isn't it? Because then you've got cells, in the following generations are all coming out the same, one on each side. But yeah, they're rather lovely. And those your possible combinations. So if you were to cross a ginger female with a ginger male, you will only get ginger kittens, all right, because they've only got ginger alleles between them. Similarly, with black female, black male, you'll only get black kittens. Because of this codominance, it means the female's got to have two black alleles, all right, the male will only have one. But if you then start mixing them, you can have your black female and ginger tom, in which case you will get black males, right? Because they can only get one X chromosome, and the only X chromosome, hang on a minute, is that right? If we've got a ginger male, it's got to give the X. Oh yeah, no, sorry, if we get a ginger X, sorry, it goes to the tortoiseshell one, doesn't it? So you can either have black males or tortoiseshell females. You can have ginger female, black male, in which case you get ginger males and tortoiseshell females. And then you've got all the other possibles as well. So yeah, if you cross your tortoiseshell with a black or ginger male, then yeah, you get quite a lot of possible outcomes there, don't you? So yeah, I rather like that it's a really, really good illustration of how these X chromosomes switch off. There are other illustrations as well. Excuse the nude lady. But this is an example of a condition which I can't remember the name of, but it's to do with the development of sweat glands. And part of the development is controlled by a gene on the X chromosome. And if if a woman has the allele for that condition, what she tends to get is this. There's a lack of development of sweat glands in some of the cells of her body, and you can actually sort of find out where it is. And I can remember seeing this illustrated on a television program once, where they used like a spray that showed up in colour if there was water. It would only need to be something simple that in a hydrated form was one colour, but in an anhydrous form was another colour. And they sprayed this woman's back, and it showed up these patches that were producing sweat and patches that weren't. And that's the sort of effect. So what you've got in this woman here is, they're showing it by The green patches are the skin areas of skin that lack the normal sweat glands, so that's where the normal X is switched off, whereas the rest, the normal skin, is where the affected X is switched off. Yeah, curious effects, and you know, it can be quite noticeable in particular areas. I suppose you could have the same sort of effect as with the tortoiseshell cats, you know, half the body sweating, the other half not, which would be very peculiar. And I think that is it on that. But yeah, I find that actually quite a fascinating thing. Did I check? Oh, I drew. Yeah, I drew on the cells, didn't I? Oh, hang on, don't save. Right. Yeah. I suddenly want to come to that and find I've got circles drawn around the cells all over the place. So sex chromosomes are actually really quite interesting, and so we've got things like, well, the other examples that go with this was the tortoiseshell cats. All right, so you'll remember that one where you've got the X, B, and X. Should call it O, but I'm going to put it G for ginger. All right, so you can either have those together, you can have two B's, you can have two G's, you can have, let's put them in, I've got a few minutes. So you've got X, if you've got two XBs, you've got your black female. If you've got XG, XG, you've got your ginger female. If you've got the XB, XG, you get your tortoiseshell. I'll put torty because I can't be bothered to write it all out. And then if you've got, for the male, you've only got the option of, whoops, I've gone from dashes to colons. So you've got your black male, and where you've got that, then you've got your ginger male. All right, so yes, it's a good example, you know, codominance there, where you're getting the effect of the X chromosome switching off. Other examples of codominance, you'd have both colours being expressed at each location, which you might get if it was something like, let's say, flower colour, and let's say you'd got a codominance between red and white. So you could have red flowers or white flowers or pink flowers. You know, you do get that sort of effect sometimes. But with this, because it's on the X chromosomes, you know, you're getting these very distinct patches where one or other of the X chromosomes get switched off. Okay, that's quite a tidy place to stop for today. The next thing I'm going to go on to will be dihybrid inheritance.